Hello everyone, welcome to a fresh session with online biology tutorials. In this video we are going to study transformation, and this is again important. Generally a 5-marker question comes on this topic. So we have already done with DNA structure. What are the components that make the DNA? What are the dimensions of DNA? Which rule is being applied on DNA being double helical? And we have solved a problem also. So if you have not watched my initial videos, please do watch them before coming to the transformation topic. So there are few experiments which we will be discussing, and these experiments are important and they will actually prove that DNA is the genetic material. This is the one that passes from one generation to the other. Along with this, there would be one experiment more that will explain, that will justify that the replication of DNA is semi-conservative in nature. So let's get started with our topic, and that's transformation. Now, if we see the general meaning of transformation means, it would be a change. Now we will see which change are we talking about. The scientist which is associated with transformation principle is Griffith. So Griffith, in the year 1928, he worked upon a microbe, and that microbe was Streptococcus pneumonia. Now this Streptococcus pneumonia could also be said as Diplococcus pneumonia. Now the species name here, pneumonia, itself must be clearing you up that this would be the microbe that would be causing the pneumonia disease, right? So here coccus word is there. I hope it must be clearer to you, because whenever the bacterium it is spherical in shape, we use the suffix as coccus. And strepto means that bacteria, they must be in chains. So Streptococcus means the bacteria, they are arranged in a long chain, and Diplococcus means again the shape will be spherical because we have used the word coccus. Diplo means two strains or two microbes will be occurring together. So here you can see that I have made 3 groups, and in each group 2 microbes are there. So diplo means 2 spherical bacteria together. Now what he did, Griffith worked upon this strain, and this strain was causing pneumonia. He used mice for his experiment. Now what he done, let's get started with it. There were 2 strains of this bacterium, right? One was the S strain and another was R strain. Now here you must be wondering, what is S and R? So in biology, whenever we abbreviate, every term has a meaning. So here R means rough strain, and here S means smooth strain. Now when am I going to say smooth? That means it must be having mucilage on it, because of which this was shiny, right? Mucilage is anything which is sticky in nature, and this smooth or S-stain bacterium, it had mucilage which gives it the shiny appearance, whereas mucilage was absent in the case of R strain. That is why it was rough in look. Another important thing, S strain was virulent in nature. Virulent means it was harmful, whereas the R strain was non-virulent in nature. That means the R strain being non-virulent, whenever it would be injected in some animal or in any of the mammalian system, it will not cause pneumonia. Whereas S strain was virulent, it was harmful, so whenever it would be injected in some animal, it is always going to cause pneumonia, right? Now we will see how Griffith made the use of these strains. So first time, what he did, he took the live R strain and he injected this in the mice. What he noticed that mice did not develop pneumonia and mice was very much alive. This was his first finding. Then later what he did, he used live S strain and he injected this in the mice, and what he found that mice was dead. Mice was dead because mice developed pneumonia, and Griffith was able to find S strains from that dead mice. Third time he used heat-killed S strain, and this strain was also injected in the mice, and after that he saw that mice was alive. So Griffith concluded that due to heating, the virulent genes, they are no more. That's why mice was alive, because it did not develop the pneumonia. So he proceeded with the fourth experiment, and that was he used this heat-killed S strain. Along with that, he also used live R strain. So what do you think the result is going to be? You can write in the comment box. I would really appreciate it. I know your answer would have been alive, but I'm really sorry. Here Griffith noticed that mice was dead, right? Individually, if you will see that R strain here was making the mice alive. Heat-killed S strain was also causing the mice alive. Now Griffith was quite confused. He was quite amazed to see the results. So what he did, he just gave a principle, and that was known as the transformation principle. You must know the definition of transformation principle as well, because when a question will come on the transformation, you need to define also what is transformation. So transformation principle, it actually says that change occurs in a genetic material by the introduction of foreign genetic material. Now Griffith was totally amazed, as I said, but one thing he did, he dissected the dead mice, and out of that dead mice he found the live S strain. This also left a question in his mind, but he was never ever able to give the name of the chemical that made this chain. So he stated that R strain was totally non-poisonous. Might be some virulent genes were still there in the S strain, and these genes went inside the genetic material of the R strain and they replicated along with the DNA of the R strain, because of which non-virulent genes were transformed into the virulent genes, and that is why R strain only got changed into the S strain. So this was the finding of Griffith. He said that the non-virulent strain was changed into the virulent state because of the transformation, because of the change. This also known as Griffith experiment, or we can also say that Griffith's principle. So if a question comes on Griffith's principle, if a question comes on the transformation principle, we would be writing whatever we have studied till now. Now, very important thing is, Griffith did his experiment, but he was never ever able to explain why this change has happened. So what happened in the year 1944, there were three scientists, you need to learn the names, MacLeod and McCarty, right? They reworked upon the transformation principle to find out the reason. So they took the heat-killed S strain and they separated the components into carbohydrates, proteins, DNA, and in one of the component they used DNA, but along with that they used an enzyme called DNase as well. So as the name is itself making a sense that ACE would be means that it would be a name of an enzyme, and this enzyme, what it is going to do, it is going to degrade the DNA. So MacLeod and McCarty, they took the culture of the R strain. Culture means whenever we grow bacteria artificially, then we use the word culture. So he took different cultures because he had 4 components at his side. So he took 4 different cultures, and then he introduced in one of the culture as proteins, in one of the culture as carbohydrates, in one of the culture as DNA, and in one of the culture with DNA along with the DNA digesting enzyme called DNase. After this, what Griffith did, he injected this in the mice. Now what they actually observed, because they all 3 worked together, so what they found is that mice was alive in this case where proteins were used. Mice was alive in this case where carbohydrates were used. Mice was alive in this case where DNA along with DNase were used. But mice was dead when this culture was used. So that means they concluded that for a change to happen, for a transformation to happen, DNA is a must requirement. It was DNA that caused the transformation of the non-virulent strain to the virulent one. That is why Griffith was able to obtain S strain from the dead mice. So the credit ultimately goes to the MacLeod and McCarty, who worked in the year 1944, and they proved that DNA is the major chemical that caused the transformation principle. So if you have not understood well, then please rewind the video and watch it again. And please don't forget to like, share and subscribe my channel. Do press bell icon for the recent notifications. Thank you.